All right, so big green button. Okay, well, thank you for the opportunity to introduce C5 Therapeutics, Carl Erickson, CEO. So I'm going to take you very quickly through this. C5 Therapeutics is developing next generation anti-C5s that we believe have transformational potential for patients with GA. I'm going to speed run through this. This is a story around de-risk science, multivalent lead program and pipeline, biomarkers for better efficacy, and a very efficient clinical path for proof of concept with the potential to launch in 2030. So complement, validated pathways systemically and in retina. And in GA, the general idea is that hyperactive complement is mediating cell death, and so inhibiting complement should reduce in less GA progression. So we're building on that theme, but really taking a page from the anti-VEGF book, where we're building more potent, longer lasting drugs and using better biology to better select patients to yield better outcomes. On the drug side, as I said, we're really learning lessons from the key translational criteria for anti-VEGFs, and I'm going to distill down 4 key features of differentiation here, which is drug design, potency, half-life, and PEG. So at a drug design level, we really think of Izervay as being the macugen in anti-VEGF terms. It's a single anti-C5 aptamer that critically has 3 unmodified nucleotides. Those are susceptible to cleavage, degradation, and loss of activity in the eye. So we're jumping directly to the Vabysmo approach with our lead program that's ready to start IND-enabling studies. And we've got 2 fully modified anti-C5s built on a bivalent molecule for greater potency and durability, and a pipeline of agents optimized for compatibility with existing long-acting technologies. Highlighting a couple key points of data, we are head-to-head more potent in a clinically validated diagnostic assay of complement deficiency and activity in human serum. In the rabbit PK model, we are twice as long-lasting as Izervay. We have about the same amount of drug in the vitreous at 2 months as Izervay reports at 1 month. On the PEG side, we've observed a consistent, robust, linear relationship between the amount of PEG per eye per month and CNV, and this is across multiple trials in both agents, and we predict our agent to be significantly below the level of PEG per eye that's associated with CNV risk. On the biomarker front, our innovation is to really think about GA in terms of 3 categories of complement. The first is hyperactive, where complement's mediating cell death. The second is homeostatic, where complement is actually maintaining retinal health. And a third category where deficiency in complement is actually driving AMD progression and GA progression. Clearly, you don't want to treat those with a complement inhibitor, and the focus is on the hyperactive patients. We have multiple lines of evidence for this across clinical trials, and I'm going to highlight for you here the phase 2 Regatta. We acquired this data set from Disc Medicines, and what we're able to show in a blinded, pre-specified analysis where we're only looking at the biomarkers, categorizing them as hyper and responsive to complement inhibition compared to those that don't, we see p-values statistically significant slowing of GA progression on the order of 50% at 6 months. Dr. Dave Brown will be presenting this at ASRS later this year and in meetings after. We believe this approach enables 2 product profiles, essentially. One is an all-comers, less doses, less safety risk. The other is a companion diagnostic, biomarker-driven, superior visual outcomes approach. And we think both of these are unlocked by the same phase 1/2. In the first portion, we are directly measuring complement inhibition in the ocular fluid through our dosing interval of 3 and 4 months with a single injection. In the second portion, using a run-in and sham fellow eye control with biomarker stratification at baseline, we expect to be able to prospectively replicate the significantly improved efficacy in the hyper-only. We believe with that data package for the right partner, this could enable at least 2 development strategies. One, a very low risk, rapid to launch, all-comers non-inferiority approach with a similar profile to the approved agents today. Or second, a companion diagnostic-based one where it's delivering significantly improved visual outcomes compared to the current products. Where we are today, we're currently having conversations here, looking forward to starting some new ones, exploring opportunities to fund clinical development of our product. I hope I've been able to communicate our story and ultimately share the excitement we have about the potential to significantly improve vision with far fewer doses, less safety risk for millions of people with GA. Thank you.
Great work, Carl.
Thanks.
I think, you and I worked on Lucentis
Yeah,
when we were treating every sort of AMD. What we've learned over the last 2 decades is that AMD is an umbrella disease, right? Not every
Exactly.
patient's going to benefit from every drug. And I like the approach of the biomarker here, but even though we want to go for the entire spectrum, but we might dilute that efficacy by not choosing the right population. And maybe just comment a little bit on what type of biomarker, but what has been the feedback from the clinical trial investigators or your advisors on utilizing that?
Yeah, so I appreciate the question. And I think, so to comment on it, the biomarker is the signature. A key component of that is ocular proteomics, so aqueous humor tap. I think the advisors are uniformly excited about the potential to pick the right patients and get efficacy. And I think it's also part of the attraction is that I think this biomarker explains some of the clinical results. People see patients getting better, but some potentially getting worse. And I think the general approach is, yeah, if you can do this, it's worth doing. And we'd also really like to find, even if it involves some dilution, a non-invasive way. Maybe we don't get as good efficacy, but we don't have that invasive biomarker. That's something we're exploring.
Yeah, congratulations. Do you know what the uptake has been so far for Syfovre and Izervay?
Yeah, I believe
Orally?
they've reported on the order of 5 to 10-ish% of eligible patients have received treatment. I think it's on the low end of that.
Yeah, it's still an open market. Thank you very much.
Thank you.